right, for this next one, we're gonna get into hearing, balance, and vision. So we're kind of covering a lot in this one. But by the time we get through this, we'll talk about and understand the gross and microscopic structures responsible for these senses. And then we'll talk about the supporting structures we find with the eye and the lacrimal apparatus as well. So we're going to really put a heavy focus on the ear and eye because the two of those are responsible for all three of these senses. So let's start with the ear, and so we're going to start from the outside and work our way in. So the external ear, I like to describe the external ear as everything you can touch with your finger. So the auricle, which is going to be the fleshy parts, this is what you think of when you think of your ear. That's going to then convey sound into your auditory canal, or what you probably know as your ear canal. And so that's why I say, like, that's what is everything you can touch with a finger, right? Because you can touch the outer part of your ear, the auricle. You can touch, at least in part of your ear canal, right? You can stick your finger in your ear. But then you can't go any further than that with your finger. At that point, at the end of the auditory canal, we get to the tympanic membrane, and that's going to be the beginning of the middle ear. The tympanic membrane is going to vibrate in response to any sound waves that hit it, and we're going to take those sound waves and then transmit them into mechanical waves. So we're taking sound and we're converting it, right? We're changing it into mechanical waves. So the tympanic membrane starts the middle ear. Then we continue that space with three little bones that we call the ossicles. The ossicles are made up of the malleus, the incus, and the stapes. And those are going to continue the conducting of those mechanical waves. We also have something called the eustachian tube, also known as the auditory tube, that connects the middle ear to the pharynx. And this is because, like, you think about what's going on in the external ear, we are filled with air, right? There's air surrounding your external ear, and there's air inside your ear canal. And in the middle ear, right, on the other side of the tympanic membrane, there's also air. But what happens if you go, like, maybe for a hike and you do a very steep climb, or you go on a plane, right? The air pressure is going to be different on one side of the tympanic membrane and the other, right? If you go very deep, or like deep in under the water or underground, or if you go, up in a plane, like high in the sky, you'll notice like you can feel that difference in the pressure. And the air pressure from one side of your ear to the other, right? The tympanic membrane being your eardrum. The eustachian tube helps equalize that pressure. So basically making sure that there is not too big of a pressure difference from one side to the other of your eardrum. And this is because if the pressure were to be very different for a while, like, it could actually rupture the eardrum. And so the eustachian tube helps prevent that from happening, right? It helps prevent that perforation, that rupturing the eardrum due to air pressure differences. And this is why, you may have noticed, like, if you go on a plane, like, your ears pop, right? Or you chew gum or something, because that tube actually connects into, like, your throat. We go past that, we get into the inner ear. This is where we have a bony labyrinth lined with a membranous labyrinth that's going to separate into different tubes and spaces. The space between the two labyrinths is filled with a fluid, and we call it the perilymph. And then inside the membranous labyrinth, we have a space filled with endolymph. And there's three separate regions in here. We have the cochlea. This is going to be responsible for hearing. The vestibule, which is responsible for balance, and the semicircular canals, which are also responsible for balance. The vestibule and semicircular canals will handle different sides of balance, with the vestibule handling gravitational balance, meaning like up and down, and semicircular canals being responsible for rotational, which would be like left and right. The inner ear is also going to be doing another transformation. So like how the outer and middle ear did a conversion from sound waves to mechanical waves, now the inner ear is taking those mechanical waves and converting it into an electrical signal. That's going to be sent through the vestibulocochlear nerve to the brain. Because remember, electrical signals, that's what the brain can understand. The brain cannot understand mechanical waves. It cannot understand sound waves, but it can understand electrical signals. And so that's the whole point. And you'll see this trend of converting something like light or mechanical waves or something along those lines to an electrical signal, so that the brain's only receiving electrical signals and being able to make sense of that. So if we take a little picture of everything going on, here's the auricle, the ear canal, the tympanic membrane, right? This is the tympanic cavity, the middle ear cavity. We see the eustachian tube, which will help equalize the ear pressure, the malleus, the incus, and the stapes. That's going to strike the oval window that the stapes is attached to, and that's what's going to allow us to communicate with the inner ear components, which are behind this bone, right? The vestibule, which is this part here, the semicircular canals, which come up here, and then the cochlea. And if you look at it, it kind of looks like a snail, right? The cochlea being the shell, the vestibule being like the snail itself, and then the semicircular canals being like the little eye stalks, right, of the snail. And then they're going to be communicating using the cochlear and the vestibular nerve, which come together to form the vestibulocochlear nerve. Now when it comes to audition or hearing, right, so audition, auditory, audio, we think hearing, noise, right, we're looking at the cochlea. It's that spiral-shaped tube, again, looks like the snail shell, and it has three compartments to it. The scala vestibuli, the scala tympani, and the cochlear duct. All of these are filled with endolymph, so we're fluid-filled now, right? We're fluid-filled in the inner ear. The stapes that's attached to the oval window is going to be right at the start of that scala vestibuli. That's going to extend right above the cochlear duct, which is like right at the at the uppermost tip of the cochlea, right? That's where the scala vestibuli curves over the top and then becomes the scala tympani, right? Then we get down to the base of it underneath the cochlear duct. The scala tympani ends at what we call the round window. Don't mix up the oval and the round window because they do different things, right? The oval window is what the stapes is actually striking, right, in order to cause waves in that endolymph. So when the ossicles vibrate, right, they hit the oval window, that's going to cause waves in the fluid inside the scala vestibuli and the scala tympani. And so when we get to the cochlear duct, we have several organs of Corti. Now this is where like hearing detection really comes in. So that wave motion in the fluid, right, in those two scala is going to be converted into neural signals when we get to the organs of Corti. So they're right on top of what we call a basilar membrane or basilar membrane, right? This is the side of the cochlear duct that's between the organs of Corti and the scala tympani. And in that organ of Corti, we have hair cells, right? They're called hair cells, not because it's made of hair, but because they have these stereocilia that look like hair extending from the cell's apical surfaces, so from the edge. And they're going to extend up to what's called the overlying tectorial membrane. So they extend up and attach, right, medially to the organ of Corti. So when these waves come through the fluid from the scala, it's going to cause movement in that basilar membrane, in the tectorial membrane. They slide across the stereocilia. That means the stereocilia bend, right? And either they're bending towards or away from the tallest member, and that's what's going to cause the electrical signal. So this, I know, it looks crazy, right? So here's like the bony cochlear wall, right? And then inside, right, we have the vestibular membrane, we have the cochlear duct, right? Here's the scala tympani, here's the scala vestibuli, and then this right here, this is the organ of Corti. Sandwiched between them, we have the tectorial membrane coming over the top, the basilar membrane coming from the bottom, and then the stereocilia, which are going to be like right here. And then as these guys, these are going to be moved over, like in response to those waves. This is just what it would look like if we were to look at an actual tissue sample. I know it's kind of grainy, so like bear with me on this. I'm not going to use this picture on like an exam because of that. But we can kind of see that organ of Corti right here, right? It looks like a grainier version of what we saw on the page before. So we look up close, right? Here's that hair cell. Here are those stereocilia that look like little hairs coming off. They're anchored to that tectorial membrane. right? And how these guys bend is going to tell us things like pitch, is it high pitch, is it low pitch, as well as volume, is it loud, is it quiet. So if we look at how sound is transmitted, so here we have sound waves, right? High and low pressure is what gives us a sound wave. And there's a wavelength, right? This is what we call the frequency, right? So the distance, distance between, or the amount of time it takes to go from one wavelength to the next, right? That's going to be our frequency. Those are going to be collected and funneled into your ear canal by the auricle into the auditory canal. They strike the tympanic membrane, which is then going to cause the ossicles to vibrate. The ossicles will amplify those vibrations, right? So the malleus, the incus, and the stapes strike that oval window, right? And then the fluid throughout here is going to move, right? It's going to move in the scala vestibuli, it's going to move in the scala tympani, the organ of Corti sandwiched between, and those hair cells are going to bend in response. That's going to tell us, right, the pressure that bends them is going to tell us frequency and volume. How often are they bending, in what direction, whatnot, and then also how far. We get to equilibrium when we're going to look at the vestibule first for balance. This is another special sense. Similar setup in terms of we're using hair cells with stereocilia as a mechanoreceptor. So these hair cells are located in the maculae of the vestibule. We have two parts to it, the utricle and the saccule. And the stereocilia of these hair cells, they extend into this gel called the otolithic membrane. And on top of which there's a layer of calcium carbonate crystal called otoliths. Right? The vestibule is responsible for acceleration of the head and gravity. So when the head moves, the otoliths move with it, and that the otolithic membrane bends the cilia in the hair cells, right? But it's also like up and down too, like, so you can give yourself, you can kind of understand the orientation of your head and whether or not you're moving. So here we're looking at utricle and the saccule here. Here we're looking inside, right inside that macula. So we see this is filled with endolymph, right inside the macula. Here we have these calcium carbonate crystals, right? The otolith and the hair cells, right in here. And so when we're just like upright, right, we see like the hair cells are just upright, right? Otoliths are suspended just straight up and down. But if we tilt our head forward, like bending down, the force of gravity pulls those down and it bends those stereocilia. So it gives us the sensation that we are now facing downward. The other side of balance is the semicircular canals, and so these ones are going to be found inside ampullae at the base of the three semicircular canals, right? So there's three of these semicircular canals, and each one have ampullae that the hair cells are found in. And the reason we have these three is because they're going to help us with different planes. So we have one for the horizontal plane, and then we have two for the vertical plane. So this is giving us kind of rotational, right, balance. The stereocilia extend to the cupula. This is a membrane that is on top of the ampulla. The semicircular canals, again, are sensing head rotation. So if we are turning left or right, we can tell from the semicircular canals. So this is what we're looking at. So here our head is still, that cupula is right up and is facing straight up, inside the ampulla. All of our little hair cells with the stereocilia are here. But if we turn, right, that cupula bends the opposite direction. So here, we're turning our head one way, and this cupula is going to bend, right, in response. It's going to give us the sensation that we are rotating, right, to some degree. We continue with the eyes and eyelids. So we'll start with the eyelids first. This is, again, vision is another special sense. We have various structures that protect the eyes since they're so vital. We are a very visually dependent species compared to other species. So we need a lot of supportive structures and protective structures. We have the bony orbits right? That'll help house the eyes. We have the eyelids and the tarsal glands, and then we have the lacrimal apparatus. The inner lid, we have a thin membrane known as the palpebral conjunctiva. The conjunctiva is what's going to connect the eyelids to the eyeball. And then at that connection, we have a thin layer, another thin layer called the ocular conjunctiva, and this is going to be continuous with the external and the anterior surfaces of the eye, except where we see the cornea. And then just superior to the white part, what we call the sclera. The conjunctiva has blood vessels and nerves. Because the sclera doesn't have any of those things themselves, we need the conjunctiva to detect foreign particles and keep them from entering the eye. So if we're looking at how we're suspended, so here's the eyeball, here's the ocular conjunctiva, right? So if you've ever had like conjunctivitis, right? Ooh, that's a protective feature to protect the sclera underneath. We have the palpebral conjunctiva here, kind of coming over the top, right, because it's part of that eyelid. Right. The levator palpebrae superioris muscle is going to be what helps like you open your eyes, like open your eyelids, right? And then here we see a little bit of some of the muscles. I'm not going to get too far into the muscles because we've already covered that. Now the lacrimal apparatus, this is going to be for lacrimal fluid, aka tears. This is so that we reduce friction, so basically we lubricate the eyes, as well as to clean the surface of the eye, so that, you know, dirt, dust, debris, things, we can blink that away, right, by washing that fluid, washing tears over the eyes. And you'll notice if something gets in your eye, you tend to tear up, right? It also helps to prevent bacterial infections because it's not just liquid. Like, it's not just like a water that makes up the tears. There's also lysozymes, there's enzymes that are antibacterial in the tears as well. So these tears are made by the lacrimal gland. These are going to be found in what's called the superolateral depression of each orbit, right? There's a little opening, right, called the superior and inferior lacrimal puncta that drain lacrimal fluid into these channels called the superior and inferior lacrimal caniculi. Right? Lacrimal fluid enters the lacrimal sac, drains in the nasolacrimal duct, delivers it to the nasal cavity or mixes with mucus. And so if you think about it, like if you cry, right, you have tears that come out onto your face, but you also might get a runny nose. And so this process, right, of using these puncta, channeling tears into the canaliculi, channeling that into the lacrimal sac, and then the nasolacrimal duct, makes sense why sometimes when you cry, you might also have a bit of a runny nose too, because some of that is tears, right? You'll notice too, like the mucus, if you get a runny nose when you're crying, it's not the same as like getting a runny nose other times, maybe because of like illness. Like sometimes it's just, you cry and it's like, it's literally like tears. So this is what we're looking at, right? So here's the lacrimal gland up here that can produce those tears. Here's the superior and inferior lacrimal punctum, right? Here's that's going to be like the little hole. And you can actually see, if you look in a mirror, especially the inferior one, you can see it if you look in a mirror. You can find that inferior lacrimal punctum. Underneath the skin and everything, that's where the canaliculi will run, right? So you can't see those because they're underneath tissue, and then the lacrimal sac here, and then the nasolacrimal duct pulling that away down towards your nose. Now in terms of movement of the eye, I'm going to go through this one pretty quickly because we've talked about muscles already, and I know you've filled some of this out in one of your labs already, so we'll go just kind of review it quickly. But these muscles that we're going to talk about are all controlled by the abducens, right? The trochlear and the ocular motor nerves. So those three nerves together are all controlling these muscles. We have the superior rectus that's going to bring the eye up, the medial rectus, which brings the eye out, so you're looking out to the side, and the inferior rectus, so you can look down. The lateral rectus will let you look in, like towards your nose. The superior oblique will allow for like kind of like if you're looking down at a corner. And then the inferior oblique will bring looking like up in a corner, like towards your forehead. So these are the muscles. I'm not going to spend much time here because we've already talked about these in lab. You can pause here if you need to go over and review where these muscles are located. So now let's get into the eye itself. So we have 3 different layers of tissue. We have fancy names, multiple names for a lot of these, but we have the fibrous tunic, the vascular tunic, and the neural tunic. You probably know these better by their other names, which would be the sclera, the choroid, and the retina. Right? That's probably how you learned them before, but we're going to learn the names fibrous, vascular, and neural tunic as well. So the fibrous tunic is going to be the sclera as well as the cornea. The cornea is like a modified section of the sclera. The vascular tunic has the choroid, which has like blood vessels and all that kind of pigment and whatnot running through it. It also includes the ciliary body and the suspensory ligaments that hold the lens in place. And it includes your iris, which is the colored part of your eye. So if you have brown eyes, blue eyes, right, that's going to be part of your vascular tunic. It also includes some smooth muscle layers, including the sphincter pupillae and the dilator pupillae, which are going to help you open and close your pupil. So if you've ever noticed, like, if the room is very bright, your pupils get small, like little pinpoints, and if the room is very dark, your pupils open up so to let more light in. So those muscles are included in the vascular tunic as well. And then the neural tunic, or the retina, that's just going to have the photoreceptors that detect light. We can also divide into 2 cavities, front and back basically, which is the anterior cavity, which is going to be between your cornea and your lens. This is filled with what's called aqueous humor, and the posterior cavity behind the lens, that's going to be what you think of when you think of the eyeball, right? That's filled with the vitreous humor. So these are all the structures. I'm going to go through a few of them, but if you need to pause to review them, please do. So again, sclera, right, is going to be part of that fibrous tunic, right? It's going to be the sclera, the white of the eye. We kind of see it wrapped around here. Cornea is part of that as well. Right? Choroid is going to be part of that vascular tunic, and then the neural tunic is going to be like what we see here in yellow, which is the retina, right? Here's the lens and the cornea, the pupil here, right here, and the iris surrounding it. This is the ciliary body, right? And the suspensory ligaments that hold that lens in place. In this space here is where we have the anterior cavity. Back here is the posterior cavity. Posterior cavity has the vitreous chamber. Now here's where it gets weird. We have the anterior cavity right here, and there is an anterior and a posterior chamber. Just basically which side of the iris are you on. Don't mix up posterior chamber and posterior cavity because those are different things. Right, so keep that in mind. We have the fovea centralis, which is this little pit where we have a lot of cones. This is going to be what we try to focus light on in the eye. And then the optic nerve, which is going to leave out here with its own blood supply. Now, because we have the optic nerve here and the way that photoreceptors are arranged, which we'll talk in a little bit, this creates a blind spot. And a blind spot is just an area that, like, you don't have any visual information coming in. You don't notice it, though, because your brain kind of, like, fills in the gaps. This is how those muscles around your iris will work so that you can dilate or shrink your pupil. And then we got to get into the real main player, which is the retina. The retina has several layers of cells that connect to each other from deep to superficial, so we're going to go in order. This is the weird part. So remember, we're going from deep to superficial, so further in the eye versus like closer to where light's coming in. The photoreceptors, which are rods and cones, are the deepest. Seems weird. The rods are involved with levels of light, so your black and white vision, but like, is it bright? Is it dim? Cones are responsible for color vision, right? Red, green, and blue, and then the combination of those is going to be what gives you the variety of colors you see. We also have horizontal cells right over the top of those, bipolar cells over the top of those, amacrine cells, and then finally retinal ganglion cells, or RGCs, which will be the ones that connect at the optic disc, right, and create the optic nerve. The center of the retina, that little area I pointed out on the slide before, is the fovea or the fovea centralis. There are no supporting cells, blood vessels. There's just photoreceptors. So we want to focus on that point. We get visual acuity, or like the sharpness of vision right there. So like focusing right there is going to be what gives us vision that is very crisp and very clear. When you're off of that, right, you're not focused on that, that's when you get blurriness. So this is what we see, and this is the weird part. So, light's coming in, here's the fovea, light comes in, the optic nerve is over the top. So light has to penetrate through the optic nerve, which is connected and created by all these ganglion cells, which are connected to these amacrine cells, which are connected to these bipolar cells, and the horizontal cells, and then finally the rods and cones at the very back. So light has to pass through all of these. So because we have this like backwards design, where the optic nerve exits the eyeball, because this nerve is passing all the way through, there are no rods and cones right here. If you're following my mouse, there are no rods and cones right here because the nerve tissue is running through there instead. And that's what's creating a blind spot. So the blind spot's where your optic nerve is connecting to your eyeball, because of this backwards design, you can't have rods and cones in this particular section, right? Again, your brain fills in the gaps, so you can find it if you use like a piece of paper and like draw a dot on it and you hold it a certain distance while covering one eye. You can find your blind spot, but you never notice it without doing something like that, because your brain is constantly filling in the gaps using both eyes, right? Because one eye can kind of compensate for the other with that blind spot. So here's that fovea, that pit, right, that we try to focus on. We see that a lot of these other cell layers are like really thin or have kind of been pushed aside so that we can have just like photoreceptors getting that light, right? So this is where we get light that hits here is going to help us with that crisp, clear image that we want to create.